Sure, sir. Assalamu alaikum. Good afternoon, everyone. I would like to thank to our honorable professor for providing us an opportunity to learn and discuss new article through this presentation event. Myself, Sabrina Mumtaz, and registration number is 910115343. Today I will present an article about synthesis of Stilbene derivatives with inhibition of SARS coronavirus replications, which was published in European Journal of Medicinal Chemistry. I have chosen this article as we already know about the recent pandemic of coronavirus and no vaccine has been discovered yet. So this article is related to the inhibition of coronavirus by Stilbene derivatives. So I will start with the background of this article. Stilbene derivatives, which are thought to be phytolexin, are widely distributed in nature. They have a wide range of activities and observed in some of the naturally occurring as well as some of the synthetic. Activities include antimicrobial, antioxidant, anti-leukemic, antiplatelet aggregative, protein tyrosine kinase inhibitory, anti-inflammatory, anti-carcinogenic activity, anti-HIV, and anti-herpes simplex virus. In an effort to find other potential activities of this kind of compounds, 17 derivatives, including resveratrol, were synthesized. 12 of them were evaluated for their antiviral potential against severe acute respiratory syndrome, SARS coronavirus, that is called, induced cytopathicity in Vero E6 cell culture. The results showed that SARS virus was totally inhibited by the compound 17 and 19 at the concentration of less than 0.5 milligram per ml, and no significant cytotoxic effect were observed in vitro. So here's some of the new term I have learned in this article, and I'm going to share it to you, is Stilbene. Stilbene is an important group of non-flavonoid phytochemicals of polyphenolic structure characterized by the presence of 1,2-diphenylethylene nucleus. Resveratrol. Resveratrol is a stilbenoid polyphenol, that is chemical name is 3,5,4' trihydroxystilbene, a type of natural phenol and phytolexin produced by grapes and wines mainly. Acute respiratory syndrome is a severe lungs condition that occurs when the fluid fills up with the air sac in lungs. Too much fluid in lungs can lower the amount of oxygen or increase the amount of carbon dioxide in bloodstream, causing the dysfunction of the lungs. Cytopathicity is effect or cytopathic effect refers to the structural changes in host cell that are caused by viral invasion. The infecting virus causes lysis of the host cell, or when the cell dies without lysis due to an inability to reproduce. Vero E6 cell culture. This cell culture are a lineage of cells used in cell culture. The Vero E6 cell lineage was isolated from kidney epithelial cells extracted from an African green monkey whose species name is Chloricebus, formerly called Sarcopithecus aethiops. This group of monkeys has been split into several different species, and SARS virus, that is our main talk, is severe acute respiratory syndrome, is a viral respiratory illness caused by the coronavirus called SARS-associated coronavirus, was first reported in Asia in February 2013. Here, I have concluded three objectives of this article is to synthesize Stilbene derivatives bearing hydroxyl group, and in some of them used pyrimidine ring in place of one benzene ring, to evaluate the antiviral potential of 12 Stilbene compounds in vitro for their inhibitory effect of SARS coronavirus, to inhibit the cytopathic effect of SARS coronavirus by the 12 Stilbene compounds in vitro. Now come to the materials and method. Here three methods has been used, is chemical methods, biological methods, and analysis or analytical method. So in chemical method is used to synthesize different Stilbene derivatives and to evaluate the activities of chemicals. In biological method, to evaluate the inhibitory effect. to inhibit cytopathic effect by infecting the cell with the SARS coronavirus. And in analytical method, there has been thin layer chromatography and different type of spectroscopic method to see the activity of the chemicals. So I will talk about the chemical method. This is mainly methoxy material was used as the starting material to prepare a variety of stilbene derivatives with hydroxyl group to avoid the oxidation of hydroxyl compounds. Introduce of one pyrimidine ring in place of one of the benzene rings for the purpose of evolution of the activity chains. Now 3,5-dimethoxybenzyl bromide, 2-chloromethyl-3,4-dimethoxypyridine or 2-chloromethyl-3,5-dimethyl-4-methoxypyridine were heated with triethyl phosphate in the presence of tetra N-butyl ammonium iodide to give the phosphates that is called Michaelis-Arbulos reaction. And then the reaction of the arylaldehyde with the anion of phosphates from in situ with sodium hydride gave the stilbene derivatives, that is compound 5 to 14 with almost they have Z isomer together with water soluble diethyl phosphate. In biological method, mainly where the virus was cultured and the viral infection in the cell culture. So the Vero E6 cell were cultured in the College of Life Science and Bioengineering Laboratory, Beijing University of Technology. And the SARS virus was provided by the Institute of Microbiology and Epidemiology, Academy of Military Medical Science. And these Vero E6 cells were inoculated in triplicate into 200 microliter of Eagle's medium containing 10% fetal calf serum in 96 wells plate. And after 2 hours of adsorption, the supernatant was discarded to remove the free virus. After 2 days of cultivation, cells were grew into a thin layer infected with SARS virus in 0.1 ml and incubated for 37 degrees centigrade, 5% carbon dioxide humidified incubator for 2 hours. And later 2x chemical series dilation in 0.2 ml medium were added to the cell well with chemical and viral control. And lastly, the cell were cultivated in the same incubator for 3 days after. And after 3 days, the cytopathic effect were recorded. Now come to the analysis. At first, the determination of the melting point, it was done in capillary tube on a Bussy-Walbath apparatus to characterize organic and inorganic compound and to ascertain their purity. Then chromatography. There has been thin layer chromatography was carried out in silica gel plate. Qualitative tandem liquid chromatography, quadruple time of flight mass spectroscopy was also done to identify and purity of the compound. And lastly, spectroscopy, there has been one hydrogen NMR spectra on Varian Innova 400 microhertz spectrometer with TMS as the internal standard, and infrared spectra on Fourier transform infrared Nicolet 20 spectrometer was also done accordingly. So this is the result of the chemical experiment. That is, I must show this is the general structure of the 12 synthetic stilbene compounds. And there has been the main compound, compound 17 and compound 19 has been shown. So the substituent group of the compound 12, 2 to 14 of stilbene derivative, basically you can see that the carbon, nitrogen, oxygen are the main molecules. And in the other part, the substituent of for compound 15 to 21. And let's focus on this another table. This is the inhibitory effect of compound 17 and 19. There has been 3 continual experiment, and this is mainly the no cytocity sign, and this is cytopathetic effect that has been shown in triple plus. And the cell was done in come to and under the virus, and there has been no cytopathic effect of these 17 and 19 compounds. And in the biological methods, we have been seen that the virus, the Vero E6 cell was infected infected by the virus. So this is the normal Vero E6 cells that was cultured under Eagle's medium containing 10% fetal cough serum, and then after the Vero E6 cell infected with the SARS coronavirus, and after 3 days cultivation, this is compound 70 which was treated with the virus, we can see, and this is compound 90 after infecting the virus, and we can see that there is no cytotoxic effect of these two cells. So the discussion come to the antiviral potential of 12 stilbene compound were evaluated in vitro for their inhibitory effect of SARS virus, and there was no cytotoxicity to Vero E6 cell in the concentration of less than 2 milligram per ml, and this means that the cells did not face any cytopathic effect of SARS coronavirus. I have concluded this through a table where I can show that the methoxystilbene, that is 7, 8, 10, 11, 14 compound has no effectivity and their concentration was 3.86, though I have talked about my previous slide that the concentration less than 2 is no effective. Then methyl ethers, that is compound 7 and 10, it inhibits 50% and their concentration was 1 milligram per ml. Compound 8, 11 and 14 that has no activity and their concentration was 2.25 milligram per ml, and finally compound 17 and 19 that whose concentration was less than 0.5 milligram per ml, and we can see that this was the effective compounds. So the future prospect of this article is, although compound 17 and 19 had one same part and compound 21 that has the same part showed no activity, it seemed that the whole molecular structure was necessary to the inhibitory activity and hydroxyl group was prior to methoxyl group. The structure activity relationship is still unclear. Further research will reveal the relationship between the substituent and the activity. But the concentration of 0.5 milligram per ml is not the terminal of the inhibition of the virus, because the research could not undergo the situation of that time. As compared with sinanserin and other bioactive compounds, which was also used to inhibit SARS coronavirus, but whose concentration was much meaningful in this study is 66 micrometer, and the result of the research, that's why not seems so good. So here is the conclusion. In early 2003, severe acute respiratory syndrome broke out in China and other countries. Many scholars and researchers were engaged in the search of anti-SARS agent and vaccine. At the same time, they used this kind of compounds to the urgent antiviral filtration in vitro. The result was exciting that the changes of the hydroxyl group site to the introduction of the nitrogen atom were beneficial to the anti-SARS activity, especially the substitute sites of the hydroxyl group in compound 17 and compound 19 has been demonstrated to be a key structure element of anti-SARS virus, and no one knows the likelihood evolution of SARS coronavirus in human and animals, but moreover the complete understanding of the pathogenesis of this virus remain tentative. In these studies, they evaluated such stilbene derivatives for their potential to inhibit this coronavirus replication for the first time and thought this discovery would be beneficial to the anti-SARS coronavirus development. Until now, this has never been reported. Thank you for your attention. Do you have any question? Sir? Hello? Screen share.
Thank you very much, Sabrina Mumtaz, for delivering a very nice and timely talk on anti-SARS coronavirus compounds, stilbenes, which are very popularly known as antioxidant compounds. So the floor is now open for asking or making comment on Sabrina Mumtaz's presentation. I am expecting some question or comment.
Hello,
from.
sir. Sir, I'm Ripa.
Yes, Ripa, please.
May I ask, sir? This is a very interesting paper to me, and also presentation was also very good.
Thank you.
I would like to ask one question. How the children work to improve our lungs, or how it is work to improve the SARS coronavirus? Hello.
Sabrina,
Yes, sir.
uh,
Okay, ma'am.
please answer
Thank you
the
so
question
much.
of
Thank you so much, ma'am, for your question. Actually, this article is based on the naturally bioactive product steel bean that is synthesized chemically in the laboratory. So the practical application of using and against the infection of SARS coronavirus has not been discovered yet.
Okay. Thank you. Thank you.
Thank you, ma'am.
any more question?
Sir, I have a question, Sir Fahana.
Yes, Farhana Sharmin.
Thank you, Sabrina, for your nice presentation. I have just only one question. Why TMS is used in NMR spectroscopy?
Okay, thank you, Apu, for your question. Okay, TMS basically chosen because of its high volatility mainly, and TMS can easily evaporated, and which is convenient for recovery of samples analyzed by in nuclear magnetic resonance spectroscopy. That's why TMS is used.
Okay, thank you.
Thank you.
TMS is internal standard for doing the NMR.
Right.
Anyway, this is a chemistry part. Thank you, Farhana Sharmin, for nice question. And next, please ask some more question. Because coronavirus is damaging and destroying our life and activity, and it is now a pandemic worldwide. So Sabrina's topic is very timely.
May I ask another question, sir?
Yes, please, Ripa.
In conclusion part, you told that children inhibit the replication of virus. So my query is, in which part of our organ it can inhibit the replication? Is it in our nose or throat or in another part? Can you tell, or is there any indication in the paper?
Yes,
Sabrina?
ma'am. Here the virus is infected with the Viro E6 cell, and they has been showed that the virus viral replication has been stopped by the synthetic steel bean, that is compound 17 and compound 19. These two compounds can inhibit the replication of the virus in the Viro E6 cell mainly. So mainly the replication of the virus in the Viro E6 cell has been stopped because of these two compounds. That has been said here.
But
Yes.
there is no any indication for human organ. Am I right?
Yes, ma'am.
Tasmina
Yes, sir.
Ripa. Actually,
Yes, sir.
the study was limited to in vitro study of Viru 6 cell, that is monkey's special
Okay.
cell line, not
Okay.
in the in vivo object, for example, rat or human.
Oh,
It was simply done whether replication in the Viru 6 cell is inhibited or not. In case of no steel beans 17, 19, they found that virus can replicate very easily. You know how virus replicate. Virus actually hijack the transcription and translation pathway of the cell, and then it make its own copy. And in case of the presence of steel beans, Sabrina showed that this replication is protected and no cell lysis or damage of the cell occurs due to the infection by the SARS coronavirus.
okay. Thank you, sir.
Any more question? Quite interesting. Sabrina.
Yes, sir.
I have a few questions. First question is, you showed the activity of steel beans against SARS coronavirus, not SARS coronavirus 2. We are now facing serious problem due to the SARS coronavirus 2, which
Yes,
results COVID-19 disease. Why?
yes, sir. Sir, can you ask me again, sir? I can't hear you properly. Your voice was broken.
My question is the biological activity that you have shown to us is against SARS coronavirus, not SARS coronavirus 2.
Two, yes, sir.
The SARS coronavirus is, the earlier version. There are three SARS coronavirus. One is MERS coronavirus, another is SARS coronavirus, and current one is SARS coronavirus 2.
Two, yes, sir.
And the Stillwin showed activity against SARS coronavirus only, not SARS coronavirus 2. Do you think that the same mechanism can be happen in case of SARS coronavirus 2?
I think, sir, further research can be taken under this article, as because there has been saying that these two compounds has the ability to inhibit SARS coronavirus. So we can predict that there have another chance to work with this virus, and we can try it in our in vitro to check whether this SARS coronavirus 2 is also inhibited by this Stilbin or by any bioreactive compounds.
Thank you very much. Yes,
Yes, sir.
we have not yet known whether these compounds would be effective against the pathogen of COVID-19. The pathogen of COVID-19 is SARS coronavirus 2. But as they showed, inhibitory activity against the replication of SARS coronavirus, there is a chance. Very good answer. You mentioned 1H NMR. It should be proton NMR. One should
Yes,
be the superscript.
hydrogen.
Proton
Yes, sir.
NMR, carbon NMR, not 1C NMR.
One. Yes,
Okay.
sir.
Another thing is that spectroscopic method like NMR is not used for determination of the activity of the compound, rather determination of the structure of the compound. Clear?
Okay, sir. Yes, sir.
Do anyone have any other question? I have another observation is that the activity of the compound, that means the active concentration was too high, milligram per ml.
Yes, sir.
Or millimolar concentration. It is too high. If you
Too high,
consider
yes.
any drug, usually it should be, micromolar or, it should be active at ppm level. That means microgram per ml. But in case of steel beans that you have shown, they showed activity, but quite high concentration. This is why not so interesting. But what we can get from this discovery, that means some structural feature is effective in inhibiting the replication of the virus, and that information may be used by the chemist and pharmacologist to design new drug candidate against SARS coronavirus 2. You should tell us something about SARS coronavirus 2. I expected that you will tell something information about SARS coronavirus 2, which is making our life hell. Thank you, Sabrina. Thank you so much for a nice presentation, and the topic is obviously very relevant to our current fight against the COVID-19. Who is the next?
Assalamu alaikum, sir.
Saiful, are you the next presenter?
Yes, sir.
Yes, please go on.
Sir, may I start?
Yes, please.
Good afternoon, everyone. At first, I would like to give thanks to my honorable course instructor for giving me a chance to present this presentation here. My presentation topics is cytotoxic dimeric quinoline terpene alkaloids from the root bark of Xanthoxylum ritsa, which is published in renowned phytochemistry journal. This is MD Saiful Islam. Here is some keywords about this paper. Alkaloids. Alkaloids are a class of
We
naturally
cannot
occurring.
see, unfortunately, we cannot see your slides. Please share the screen. Saiful.
Sir, I am trying.
Yeah, please open your PowerPoint file in your laptop, and then go at the bottom. You can see share screen, and then you can find it and double click, and then it will open. We need to see your slides.
Yes, sir. Sir, here screen sharing has failed to start. Please try again later. Error code দেখাচ্ছে স্যার